Hi, my name is Rebecca Torres, and this presentation is for medical biochemistry at the University of New England. The information presented in case number 3 suggests that the 5-year-old patient has type 1 diabetes. The patient's baseline serum insulin level is 0.8, which is below the control level, and his serum insulin level 1 hour after glucose load is 5, also below the control level. In a healthy patient, as pictured here, upon consumption of food, our body absorbs glucose into the bloodstream via the intestinal tract here. This increase in blood glucose levels then triggers an increase in insulin levels in the blood, which helps to maintain a normal blood glucose level. The patient presented here, and also in our case, maintains a low blood insulin level, both at baseline and upon glucose consumption. This suggests that the patient is not producing sufficient amounts of insulin even upon consumption of glucose, which would normally increase insulin levels in a healthy person. The patient also shows signs associated with hyperglycemia, such as persistent thirst and frequent urination, which are common amongst type 1 diabetic patients because their kidneys are working overtime to filter the excess glucose in the blood. I would predict that the patient's fasted blood glucose levels would be above normal range because there is not enough insulin to regulate the blood sugar levels. Insulin would normally work by enhancing transport of the glucose consumed into tissues, thus away from the bloodstream. However, in this patient's case, there is not enough insulin to enhance the transportation of glucose into tissues, so the blood glucose levels would remain at high levels in the blood, causing hyperglycemia. As you can see here, the plasma glucose levels of a diabetic patient are raised as compared to a normal patient. This is due to the lack of insulin regulating the blood glucose levels. 2 regulatory enzymes that are likely to be active in this individual's current condition are protein kinase A and fructose 2,6 bisphosphatase. Insulin, as you can see here, normally activates phosphodiesterase, which cleaves cyclic AMP or CAMP, a second messenger. Since there are low levels of insulin, cyclic AMP levels will increase, and this increase in cyclic AMP levels will activate protein kinase A, which activates the enzyme fructose 2,6 bisphosphatase and lowers levels of fructose 2,6 bisphosphate. You can see here, cyclic AMP activates the protein-dependent kinase, which activates fructose 2,6 bisphosphatase. The reason that I do not suspect that the patient has type 2 diabetes is because if the patient in fact did have type 2 diabetes, then their blood insulin measurements would be at normal levels. In a patient with type 2 diabetes, they have an impaired sensitivity of tissues to insulin's actions, meaning that they produce enough insulin, so there would be plenty of insulin in their bloodstream. The reason for hyperglycemia in individuals with type 2 diabetes is due to cells failing to respond to insulin properly. As you can see here, in a healthy patient, insulin is produced, binds to the insulin receptor, and that allows glucose uptake into tissues. In a patient with type 1 diabetes, as in our case here, the pancreas fails to produce enough insulin, so nothing is actually interacting with the insulin receptor. In type 2 diabetes, enough insulin is being produced, meaning that the insulin blood levels are normal. However, the insulin receptor is what's failing to respond properly. Additionally, a reason why the patient's fasted blood glucose levels would not be below normal range is because the uptake of glucose into tissues and away from the blood is impaired, thus leading to an increase in blood glucose levels. The patient would only display blood glucose levels below normal range if they had been administered too much insulin and not enough glucose into their bloodstream. Now on to case number 4. Based on the symptoms and tests of the 2-month-old female presented, the most likely diagnosis is glycogen storage disease. There are multiple indicators that the infant has glycogen storage disease, and they are as follows. During the fasted state, our blood glucose levels are maintained through glycogenolysis in the liver. During glycogenolysis, stored glycogen is broken down to glucose, which is released to the blood to help maintain blood glucose levels. In individuals with glycogen storage disease, they are unable to use glycogen due to a deficiency in the enzyme needed to convert glucose 6-phosphate into glucose. This enzyme is called glucose 6-phosphatase. Since there is a defect in this enzyme in individuals with glycogen storage disease, they are unable to maintain normal blood glucose levels in the fasted state, thus leading to episodes of hypoglycemia. In addition, since individuals with glycogen storage disease are unable to convert glycogen to glucose and glucose 6-phosphate into glucose, this glycogen builds up in the liver, leading to hepatomegaly or an enlarged liver. The symptoms presented in this particular case also support the suspected diagnosis of glycogen storage disease in the case when epinephrine is administered. As you can see here, in an individual without this disease, epinephrine would activate cyclic AMP, which would then activate glycogen phosphorylase, and thus glycogenolysis, the breakdown of glycogen to glucose 6-phosphate and further to glucose. In an individual with glycogen storage disease, the pathway for glycogenolysis works normally up until we reach glucose 6-phosphate. With a deficiency in the enzyme needed to break down glucose 6-phosphate to glucose 6-phosphate builds up, leading to an accumulation of it. Now, if you recall, glucose 6-phosphate is also used in both the pathway of glycolysis and the pentose phosphate pathway. When glucose 6-phosphate passes through glycolysis, its end product is pyruvate. Pyruvate then can either produce lactate in the absence of oxygen or continue on to pass through the citric acid cycle for the production of ATP. The administration of epinephrine causes an increase in glycogenolysis, thus an increase in glucose 6-phosphate and an abnormal flux through the glycolytic pathway, leading to an increased production of pyruvate and thus an increased production of lactate. The reason that the infant has normal blood glucose levels in the fed state immediately after breastfeeding is because the glucose from the breast milk is maintaining normal blood glucose levels, however, only temporarily. The reasons why the other 3 answers are incorrect are as follows. The infant has only ingested breast milk since birth. Breast milk consists of galactose and glucose. This means that even if the infant had hereditary fructose intolerance, we would not be seeing these symptoms since she has not consumed any fructose in her lifetime. If the infant had galactosemia, then there would not be any effect on glycogenolysis during the fasted state. In individuals with galactosemia, there is a deficiency in galactose 1-phosphate uridyl transferase, leading to an accumulation of galactose 1-phosphate in the liver. An individual with galactosemia can still break down glycogen reserves into glucose to maintain blood glucose levels in the fasted state, since the enzymes necessary for this process are unaffected. Lactate would not accumulate because there would not be an accumulation of glucose 6-phosphate, and the glycolytic pathway would not be receiving an abnormal flux of glucose 6-phosphate. Lastly, the infant would likely not have glucose 6-phosphate dehydrogenase deficiency because an impairment in this enzyme would have an effect on red blood cells within an individual, causing hemolytic anemia and jaundice, which is not evident in this patient.